Good morning, everyone. So we're going to start the second day of the conference. Before we start, we are still missing a lot of the presentations for after the break. So if you're one of the presenters, please send by email to Alexis or bring it by flash drive here during the break. And so our first keynote will be this morning will be Sara Mathieson. Sara is assistant professor at the Haverford College in computer science. Prior to that, she was a PhD student at UC Berkeley. She was really a pioneer in machine learning for evolutionary genomics. She did a 2016 paper, I think, on deep learning for population genetic inference, and also I think you were involved in a 2018 paper on explainable neural networks. It was one of the first accounting for invariance in alignments. So that's yeah, she really did some pioneering work in this field, and I think that now she moved more to she focused more on generative methods, and that's what you're going to talk about this morning. Thanks a lot.
Thank you so much to the organizers. It's really wonderful to be here and have a whole conference about machine learning. So today, as Laurent mentioned, I'm going to talk about GANs in particular, and also how we might think about interpreting these types of kind of more complex machine learning methods. So in 2018, yeah, the first CNNs came out for population genetic inference. And I think since then, that has become one of the main tools for different types of inference. And I think one of the motivations for this, and the reasons I think these have been successful, is the filters that pick up on these local patterns in population genetic data along the sites or along, say, sorted haplotypes. So now, whatever your question of interest, there's probably a CNN out there that does it. So if you're interested in selection or admixture, introgression, pairwise TMRCA inference, there's probably a CNN that does that. And if there's not, you can probably build one pretty easily. So I think they've become one of the sort of standard methodologies in population genetic inference. And I think that it's almost difficult to keep up with all the literature now. So if you can't keep up with all the CNN papers or all the machine learning and population genetics papers, you can also just try to keep up with the reviews. So these are all the reviews that I found recently about population genetic inference. And then I would definitely recommend this one by Kevin and Matteo, and this one as well by Barak and Flora, who are here. And I think they kind of give an overview of what types of methods are out there. And actually, in this paper by Kevin and Matteo, they highlighted these three areas: interpretability, robustness, and representation of population genetic data, that are areas that we need to work on as a field. And today I'll talk more about interpretability and also about generative models, but I think that's something that we should kind of continue to focus on as a field. So, why I don't need to tell this audience that deep learning is increasingly generative. And this was just some examples from GitHub Copilot that I tried. So first, I just gave it this prompt, compute pi. This is pairwise heterozygosity among the number of pairwise differences among haplotypes. And I don't know, it doesn't make so much sense, but I tried again and I gave it a little bit more for a prompt. And actually this does correctly compute pi, although there are more efficient ways of doing this. But all this is to say that I have been and continue to be very inspired by generative approaches and other deep learning approaches in other fields, such as images or natural language. And I think that it has so much to offer population genetics and other types of biological data as well. So why do we care about generating data? Like, why do we want synthetic data? Why do we want fake data? Well, really, it's in all of our work. We are always looking at simulated data to validate our methods because we don't really have very good real data that's labeled or has some sort of knowledge of its evolutionary history. And increasingly, we use it for training machine learning models. And just to say that I think that the quality of the simulations we use is really important. For example, this is a few examples from different populations. This is data from an Amish population in Pennsylvania. This was the real IBD links. And then I tried actually very hard to match this in simulation, and I really couldn't really match the properties of this real data. Another example from an African population where I used sort of the defaults in standard PopSim, which is kind of built upon MS Prime, and also got a pretty poor match between the simulated and the real data. And so then if we train our methods on the simulated data, and then we plug in the real data as our test data, then we're not really going to be very confident about our inferences. So I think the match between the simulations and the real data needs to be more faithful. So today I'm going to talk about our method pggan for creating such simulations, and then how we can actually use parts of that network to really think about more local types of inference, like selection along the genome, and then finally talk about interpretability. And please, you can stop me throughout for any questions. Let me know. Okay, so you may be familiar with GANs, but just to say it again, GANs really have two networks, usually neural networks, but they don't have to be, but they are kind of working against each other. The generator is trying to create sort of synthetic data, and this is often akin to this art forger trying to create high quality simulations or fake artwork. And then you have the discriminator, or this art critic, which is trying to find out the match between the fake and the real data. And I think the key thing to understand about GANs is this idea of improvement. So the generator starts off with just random fake data. It doesn't match the real data at all. And then this discriminator is easily able to tell, okay, this is a fake data, this is a real data. But that feedback then helps the generator create better and better synthetic data. And then at the same time, the discriminator is forced to learn very subtle differences between the real data and the simulations. And then over time, they both get better. And by the end, hopefully, the discriminator is actually confused because the simulated data is so high quality. So that's the sort of goal it's been very successful in things like images. So I had an example of that so this is actually text prompt. So that's the input. So say this bird is red and brown in color with a stubby beak. And these are different GAN methods that actually try to create the synthetic data, and they're pretty good. Right now, GANs with faces, with sort of different types of images, it's indistinguishable from reality. So like a human cannot tell that it's generated by a AI or not. So for our type of GAN method, the discriminator is very similar in the sense that it's just binary classification. So the idea is you have your generated data, and then the real data, and the discriminator is really just trying to make this determination: is it real or is it fake? The generator is actually based on an evolutionary model in our case. And the idea here is that you would parameterize for us, demographic model, but you could make this more flexible with population sizes. So say there's an ancestral population size, and then two subpopulations that split, and then maybe some migration. So you could make this as complicated as you want, but some demographic model. And the generator is actually learning the parameters for this demographic model. And then whatever parameters create data that's indistinguishable from the real data, those would be the best demographic parameters. We use a variety of different demographic models, and we actually train this through simulated annealing because there's not a differentiable, this generator is not differentiable. But I wanted to mention there's a recent paper from Graham Gower and Fernando Racimo about making this a little bit different. So you can actually do a kernel density estimation for learning the parameters. But I'd encourage you to think about this, because I think that this issue of having a differentiable generator is a problem for our method and something that people could work on or explore further. So these were all the different demographic models that we used in our paper, but it is flexible in the sense that you could define a new one and plug in whatever you want for these demographic parameters. I also wanted to mention that there are lots of GANs without evolutionary generators, and these are often created for privacy reasons, where you need to have a closer match between, say, the SNPs of your data. So encourage you to check these out as well. Yeah. The discriminator, so that was the generator. The discriminator architecture is as a convolutional neural network, and this is more similar to images, but we actually use a permutation invariant neural network for the CNN discriminator, and this has a number of haplotypes and then a number of SNPs on the columns, and then The convolutional filters are actually just one-dimensional, so they slide along the sites, but not the individuals. And then we collapse all this information after several convolutional layers through some permutation variant function. It could be max or sum or average or something like that. So in this way, you get the same, kind of like some of the talks yesterday, you get the same output no matter what the order of your haplotypes are for this type of neural network. We also extended it to multiple populations, so say up to 3 populations. You could extend it further if you wanted to. The idea here is that the haplotypes are permutation and variant within each population, but then between populations, they're not. So you can kind of create this architecture that basically expands this notion of permutation and variant CNN, but not between the populations. Okay. So yeah, and this didn't come out. I'm going to skip. There's no math slide for this talk, yeah. But I did want to talk a little bit about what can go wrong in GAN training. So GANs are sort of notoriously difficult to train. They have a lot of problems with the generator and the discriminator learning at different rates. And although there are some ways around that. So this is an example of failed training. So this is the training iteration on the x-axis, and then this is the accuracy. And what's happening here is that at some point, the discriminator is classifying everything as real data. So you get 100% accuracy on the real data, but 0% accuracy on the fake data. So actually, if you average this, you get 50% accuracy for the discriminator, which looks great. Oh, the discriminator is totally confused. But you really have to separate it by your 2 classes, real and fake, to know what's going on. And then basically, because there's no feedback, it's kind of like everything gets into the Louvre, and then there's no motivation to create better artwork. So this is something that is really difficult to come back from, and basically your GAN training just degenerates. GANs are also very difficult to evaluate. But one way that we tried is through summary statistics. And in this case, it's not super important what all of these are, but this idea that your simulations in gray are different than the real data in orange here. And so this is the results of that failed GAN training, is that there's a big mismatch between your real and your simulated data. On the flip side, what does a successful GAN training run look like? So it starts off usually with the discriminator able to tell the difference between this real and simulated data easily. So you get basically 100% accuracy. And then over time, it gets a little bit more variable, and the accuracy on fake data and real data sort of more approaches 50%. And you can also tell this from the loss functions, that the generator and the discriminator have roughly the same value for their loss functions. They're kind of balanced. They're equally sort of pitted against each other. So this is what a successful training run would look like. And in this case, there's a very close match between the simulations and the real data. Any questions so far? So I'll show just a few results for this because I want to talk about the project after this. But this is an example of, this is the DADDY model of 3 populations out of Africa event. And these numbers that we get from PGGAN pretty closely match a lot of the other literature, and also create simulations that match in terms of summary statistics. And I should say that this is not conclusive that our data is, creating high quality simulations. But if we're not matching summary statistics, we know that the data is a poor match for simulations. So these were the 3 populations, and then what we noticed is that overall, there's a lot of good, we are creating high quality simulations, but there's a few cases where there's kind of a mismatch. And in particular, Tajima's D, which is often used to detect natural selection, there's kind of a poor quality match here. And we were wondering if that could maybe actually be natural selection, because our simulations do not include any natural selection, they're neutral. So this led us to this idea of trying to think a little bit more about how we could use the discriminator to identify kind of outlier regions, or something that was a little bit unusual in our data. And one thing to think about in terms of motivation for this is that often people train a GAN, right? And then they think about just using the generator to create synthetic data, right? and they kind of throw away the discriminator. But the discriminator actually contains a lot of information about the structure of data, what makes sort of something look very realistic and or close to your real data, and what makes something look more like a simulated data. So we actually kind of throw away the generator and then use a discriminator to actually identify these outlier regions. So that was the motivation for this project. So we take these trained CNN discriminators and use them in a transfer learning framework to understand natural selection. So just to kind of summarize that, if we see something that has a really high probability of being real, we think there's also a likelihood that it's likely to have some process that wasn't in our simulations. That could be mutation rate variation, unusual SNP density, structural variation, some type of selection. So we're kind of looking for those regions where there's a very high probability of them being real. If there's a very high probability of them being simulated, then they probably are a little bit more neutral or sort of evolving under kind of the standard process for the genome. So that's what we did. We threw away this discriminator. And then what we did is instead of having this be the probability of real as our output, we changed that to be the probability of selection. And then the other motivation for this is that selection simulations are very slow. So if there's a way to train a model to detect selection without actually simulating selection, then that would be very advantageous. So most of the training for this method actually occurs with neutral simulations. And then what we decided to do is sort of further incentivize the probability of selection being high for selected regions with a small number of selection simulations. So that's what we did. And we simulated these with SLIM. And then we basically feed these into an already trained discriminator as a fine-tuning or transfer learning step. But you could do this with any type of simulations for selection or any sort of non-neutral processes that you're interested in. So now the neutral label is 0 and the selection label is 1, whereas before it was simulated data is 0 and real data is 1. So that's the idea. So the kind of whole workflow of this is that we train a lot of discriminators with the regular PGGAN approach. There's no selection simulations. It's just neutral processes. And then we actually do this discriminator evaluation step just to make sure that we're not having any discriminators that were poorly trained, and we discard them. And I think this was around like maybe 5% or 10% of them. So it's not a huge number, but we do discard them if they haven't learned anything. And then we fine tune the successful discriminators with a small number of these selection simulations. And then finally, after all of that, you could plug in a different data set. So a data set that wasn't used for training. This could be from a different population or simulated test data from another situation, and evaluate the performance. And this is on GitHub if you're interested. And I think it's fairly usable at this point. And I'm also happy to share the trained discriminator networks. There's CNNs that can actually be repurposed for a lot of different things. And I pretty much know that they work. So if you are interested in them, I'm happy to share those trained networks with you. Okay, so how did this do? So the data sets that we evaluated on, we wanted to have kind of a training population and a testing population. So we chose 2 European populations, 2 African populations, and 2 East Asian populations. And we train on one population and then we test on the other sort of similar population. And then in terms of the sample sizes, because we're using a permutation invariant CNN for the discriminator, the training set size doesn't have to match the testing set size in terms of the number of individuals. So this was all of our data. And then we actually wanted to use both simulations and real data to evaluate our method. And that's a little bit difficult because we don't actually know exactly what was under selection. But we use these selected regions from this Grossman et al. Paper. And then these are pretty solid regions that we think are under selection in humans. And then we decided to put those in as kind of a real truth set. So that's how we're able to get some like raw curves for real data, which we normally couldn't do. So the dotted line here are before the fine tuning. So this is just with the regular PGGAN GAN training. And so already we see that there's a little bit of signal that regions that have high probability of being real are also high probability of being under selection. So there's already a little bit of signal, but then after fine tuning, we actually get quite a bit more. So these curves just basically shift upward, and we're able to kind of refine our signal of selection in these cases. So we were overall pretty happy with this. And we also then kind of analyze these in a little bit more detail by breaking it down with the selection coefficient. So anything with like a slightly stronger selection coefficient, basically the discriminator prediction was, it's 100% correct. It's under selection. And then for real data, we compared, say for this example, the neutral regions from GBR, which is a European population, and as opposed to positive selection. And this is, again, our discriminator prediction. And we also find that we get a lot sort of higher discriminator predictions, meaning they should be, that's basically the probability of being under selection, that those kind of match with things that are likely to be under selection, and then things that we know have a very strong selection coefficient. And then finally, we did a whole genome scan with all three of our test populations. And then we found some known signals of selection like lactase and things that affect different pigmentation, and also known signals in African populations as well. And then these we think are potentially novel targets of selection. We're doing some downstream analysis to try to verify those as well. One thing that could happen, we found these regions here that have a really high probability of being under selection, at least according to our method, but there doesn't seem to be any genes or other known things going on there. So there could be something else. And I think that's where this idea of interpretability would come in, is how do we know that our method is not just picking up on some region of low mutation rate or something like that? So we kind of need to think about some of these regions that don't really show up when we look at known gene. And then the last part of this section, we also tried other types of selection, so adaptive introgression and balancing selection. Balancing selection worked really well. It seemed to really respond to the signal. Adaptive introgression, not as much. And I think there's maybe just not as much signal about that in the region sizes that we were looking at. So this is, I think, something we need to think a little bit more about. One other question that came up was, what if you did the fine tuning step, but with a non-PGGAN trained discriminator? So just a randomly initialized network, and then just tried to do only the fine tuning step. So one thing we found from that, so this is randomly initialized versus GAN training, and they approach roughly the same loss function, but at different speeds. So there's a time component there. But also one thing we found is that, so these are the before fine tuning here with selection simulations. So they're just random. It's just random predictions. But we noticed that the simulated data after that fine tuning step, it actually did pick up on a lot of signal of selection. But for the real data, this was quite a bit lower AUC than when we actually did the PGGAN training. And my takeaway from all of this was that if you train with only simulated data, then basically your method will really only work on simulated data. So it actually kind of had the same performance for simulations if you just started from sort of a randomly initialized network. But on the real data, it was really not able to kind of understand the subtleties of real data that's built up over the GAN training. So I think this is something for us to think about as a field too, because often we're evaluating on simulations, and I think, if they're exactly the same as what we trained on, then probably it will work. But if there's some way of validating with real data, then I think that the real data needs to be part of the training process there. Okay, so the last part is trying to think about, how do we actually interpret these types of methods, or what can we learn? What can we learn from a CNN discriminator? So I wanted to mention a little bit from image interpretability, which people may have studied or may not be aware of. But I think there's kind of a misconception sometimes that all neural network methods are black boxes or not interpretable. And I think for images in particular, that's really not true. And not to say it's a solved problem, but there's lots and lots of ways of understanding why a method might say that this is a dog. So, and then in this case, just to kind of talk through this a little bit, there was high probabilities for electric guitar, acoustic guitar, and Labrador in this photo. And then these are explanations. So this is the part of the image that this method focused on when it said that it was an electric guitar. And I don't know a lot about guitars, but it seems to kind of make sense to me. And then this is the part of the image that was focused on when the method said this was an acoustic guitar. And then the Labrador or the dog, it focused on the face. So it does make some intuitive sense that the pixels of the image that are being most focused on for these different classifications kind of make sense with our intuition. So there's a lot of different ways you can do that with images. And I think that again, it's not a solved problem, but I think it's kind of it's a very mature field. In population genetics, I think we're getting there with interpretability of machine learning methods, but still kind of in the earlier phases. And I think a lot of the previous work on interpretability has focused on kind of translating some of the image methods into a population genetic space. And actually, both these methods use saliency maps, which is a technique for understanding which are the most important portions of the data when making a certain classification. And I think they do lend a lot to understanding. But one thing to note is that because, say, this was an adaptive introgression paper, and so they sorted the haplotypes based on their similarity to Neanderthal. So it makes sense that you're focusing more on this region because these European haplotypes are most similar to Neanderthal sort of by design. So sometimes the pre-processing that you're doing can kind of incentivize certain types of interpretability. In this work, the different features that were input to the method, we're looking at TMRCA, and this was the focal site and the receptive field. And this also, I think, makes sense because you're looking around, around this site, but some statistics are more important than others. And that also kind of gives you a lot of insight, like this XOR, and IBS was important kind of right around there. So they kind of lend some intuition for what the method is doing. So saliency maps and also SHAP values, which are another technique, kind of different method, but like similar ideas that you're trying to focus on the parts of the image that are most important. So these, in this paper, they were looking at neutral regions versus selective sweeps, and they also sorted them again by haplotype frequency. So this is the most common haplotype here. And then you can see that in the SHAP value, this is the area that the network is focusing on. So it's kind of, again, like pre-processing is kind of incentivizing you to focus on certain parts of the image. And you can still interpret that, and that makes sense. But I think that we need to kind of think a little bit more deeply about what methods might work for population genetic data specifically. So what we tried to do is first investigate whether the neural network for the discriminator was actually kind of computing summary statistics while that we know to be informative, but it was sort of learning to compute them on its own. And so I looked at a bunch of different summary statistics like site frequency spectrum, these inter-SNP distances, measure SNP density basically, linkage disequilibrium, pi, which is pairwise heterozygosity, and just the number of haplotypes. And one thing we noticed across the board was that pi seemed to be highly correlated with a lot of, so yeah, I should say, these are the hidden units in the layer right before the final classification. So after the convolutional layers, they're the output of the fully connected layer right before you go into the output. So in theory, these should have distilled down as much information as you can from this data. It should be like the most distilled information. So we found that pi was very highly correlated with a lot of these hidden units, as well as sometimes inter-SNP distances and LD. But it wasn't always the same for different discriminators. This was a CEU trained discriminator and a YRI trained discriminator. But we're noticing that some don't seem to be correlated with anything. Some of these hidden units. So what are they doing? We're still kind of unclear on what this means, but we think that there's some correlation with known summary statistics. What we also try to do is just look at the raw output of the network. So this would just be the probability of real or probability of selection after fine tuning. And one thing we found was that it does look very different for neutral regions versus selected regions. So for neutral regions, we found that pi was again negatively in this case, but strongly correlated with the output. So maybe the very final number that the discriminator is computing is some computation of pi. And then for selected regions, we found that this is the number of singletons in the data, which we know is informative for selection. And then intercept distances were a little bit more important. But then also you kind of see this LD pattern where these are SNPs that are closer together. And then these are the patterns for SNPs that are further apart. So it seems like both short range LD and long range LD are important, but kind of in opposite directions, which I think also makes sense that around the selected site, you see very sort of hitchhiking and correlation, but then far away, you start to see that kind of pattern break down. So we thought this was pretty interesting and maybe a future direction to look at other summary statistics as well. And then lastly, what we tried to do, I was also really interested in looking at the convolutional filters themselves. So this is just the architecture again, but I kind of basically like cut the network here and I looked at after the convolutional filters and the permutation invariant function, what filters were sort of turned on, and very few were. So there were 64 filters in this last step right before you go into the fully connected layers, and only 3 of them for this particular discriminator, but for many of them in general, only 3 of them were actually non-zero after ReLU. And so it's kind of curious what was going on here. So what I did, so these are the filter 16, 29, and 51, and these numbers don't mean anything, but just there are 64 filters total in this layer, and these 3 had, after the data was fed through, these were the only 3 that had non-zero output, basically. And so I first looked at just whether they were correlated with generally the summary statistics and saw, again, very highly correlated with pi. And then I looked kind of along the genome. So this is pi along the genomic region that we're looking at. And then these are the different filters. And it seems like they're actually kind of computing pi locally along the region. That's my interpretation anyway of what's going on. How might they be doing this? So one question is, are they looking at all pairs? Well, I don't think so. There's nothing in the network that would incentivize them to do that. And computationally, I don't think that's what they're doing. But there are other ways of computing pi just by the number of minor alleles, the minor allele frequency and the major allele frequency. And so that's how I think that they're computing this. And then that would be linear in the number of sequences and the number of sites, which I think is probably more reasonable given the way that the filters are being slid along everything. There's nothing else that could be happening, I think. And then I also looked at the filters themselves, and this is just, very, very recent work, but I don't know totally what to make of this, but this would be sort of along the genome, along that genomic region. And I think that there's some patterns in these filters that are maybe suggesting it's sort of picking up on either kind of patterns along the genome or different correlations and progressing them to the next layer. So I think this is definitely something I'm interested in, but I don't have all the answers yet. Yeah. And so a few kind of concluding thoughts about all of this. I think that there's a lot of different directions we could go in terms of interpretability. And I think that one thing I've talked about mostly today is global interpretability. So what is the architecture of the network doing as a whole? What is the behavior of it as a whole? But I think local interpretability is also really important. So why is it making a prediction that this specific region is under selection? That I know, I don't know. I have less intuition about. And then one thing I'm interested as well is we've looked at statistics that are more traditional, like SFS and pi, but what about tree-based statistics? Are they learning, because that's been also common to plug into CNN methods, and maybe they're learning novel statistics or using other approaches to get at this information. In terms of different architecture choices, I think there's a lot of things we need to think about in terms of the relationship between our data representation, architecture, and interpretability. So I think the way that we actually put the data into these methods can help us make the method more interpretable or less interpretable. However, if we do too much pre-processing, we're kind of losing maybe some of the inherent information in the data and moving more back towards summary statistic approaches. So how can we kind of think about letting the method progress on its own while also guiding it toward things that we know are biologically realistic, and that will help us be able to interpret the method as well. So I think that's kind of a trade-off there. And different architectures, different neural network architectures are just going to be more or less interpretable. If we simplify them, as we saw yesterday, you know, they might be easier to interpret. And if they're too complicated, it's going to be naturally a more difficult problem. And I think in general, one thing that I've seen recently is a lot of interesting work on simulation misspecification. If you haven't seen this data CM method from Mo and Siepel, I'd really encourage you to check that out. It's another way, kind of not like again, but it's addressing the question of how do we create the best sort of match between our simulations and our real data, and if that match is not very good, how can we correct for that? And then I think also a lot of these methods, myself included, you know, work on really clean, real data sets. But I think that there's a lot of work that needs to be done in terms of translating these methods into non-model species. And I think that this could be, this may also force us to make our methods more robust, which I think often, especially for GANs and other types of really complicated methods, the training of them is so difficult that they're not really very easily accessible to people who want to just try them out for their data set. And with that, I wanted to thank my group and funding, and thank you for your attention.